All right, so hi, my name is Megan Williams, and today I'll be discussing my study that focused on the impact of elevated carbon dioxide levels on weed growth in the C3 weed Chenopodium album, which is more commonly known as common lance quarters, and the C4 weed Amaranthus palmeri, which is more known as Palmer amaranth or pigweed. So the main objective of the study was to understand the physiological response of these weeds to elevated CO2 conditions, so we can better understand how the differences in carbon dioxide uptake and use in C3 and C4 weeds will react in the face of steadily rising carbon dioxide levels in our atmosphere. These two weeds were chosen because of their similar biological traits, such as emergence time, growth pattern, and seed type, all which lead to these weeds being really competitive with crops and really hard to eradicate and to prevent their spread. So for some context, weeds are extremely costly to agriculture, with weeds accounting for between $7 and $10 billion in losses each year, approximately. And with changing climate making our environment even less hospitable to agriculture, we need to know how our current threats are going to react to that. So as you can see from this picture of me in the field, when left unchecked, these weeds can quickly take over entire fields. This year, atmospheric CO2 levels reached 420 parts per million, when in pre-industrial levels are about 270 parts per million. This is a lot of change there. And depending on how carbon emissions are managed, carbon dioxide levels could reach up to 1,000 parts per million by 2100 in the absolute worst case scenario. And even the best case scenario, levels will continue to rise. So in order to test how Palmer amaranth and lamb's quarters react to elevated carbon dioxide levels, I tested each species at 3 different exposure levels using 3 different controlled growth chambers. The plants were grown from a frozen seed stock that was then treated with gibberellic acid to promote germination and then germinated in a growth chamber. They used potting soil to make sure they had plentiful and equal nutrient availability. The pots were kept in plastic trays to allow for bottom watering. And once they were processed, I used a leaf area meter, like the one pictured, a lab oven, and a scale to collect data. So once they're placed in the growth chambers, 2 growth chambers received elevated CO2 conditions, while the third was control and subject to just the ambient levels of the growth chamber room. Each chamber received the same daylight hours, temperature, and watering to keep that consistent. And every 7 days for 5 weeks, I removed 13 plants of each species from each growth chamber for processing. I found the total leaf area of each plant using the leaf area machine I showed, and the leaves and stem of each plant were recombined and dried in a lab oven and then weighed to find the total above ground biomass. I recorded the CO2 levels every day, and the control chamber averaged around 475 parts per million. The mid-elevation chamber averaged around 602 parts per million, even though the chamber was set to 700 parts per million, and the highest chamber averaged about 676, despite being set to 900 parts per million. Seems like the machines just weren't able to keep up with the demand for carbon dioxide. Ideally, I wanted the set levels to have a larger difference in response, so we can see a greater response to differences in CO2 level. But a test showed that over the first couple of weeks, there is not a significant difference between the 2 elevated CO2 treatments. And therefore, basically, in the next graphs, you'll see that it still includes both elevated CO2 levels, but there was not a significant difference really between the 2 treatments. So before beginning this experiment, and based on available knowledge about Palmer amaranth and lamb's quarters, I predicted that under elevated CO2 conditions compared to the control, both Palmer and lamb's quarters, leaf area, and biomass would increase. I also predicted that due to differences in C3 and C4 plant function, lamb's quarter, leaf area, and biomass would increase more compared to Palmer amaranth. Before we get to the findings, I just want to show you some pictures of what the plants looks like during the experiment. These pictures show their voracious growth over just 2 weeks between week 3 and week 4. This is what makes them such strong competitors in the field. They nearly doubled their size. You can see the Palmer amaranth on the left of the group and the lamb's quarters on the right. Here, this is week 4. This is the control chamber of both Palmer and lamb's quarters in here. This was the medium elevation of CO2, and this was the highest elevation of CO2. So you can see medium and high approximately similar, but a much lower growth rate with the non-elevated CO2. So this is for above ground biomass. A two-way ANOVA test showed that there was not significant interaction between the species and the CO2 level for 4 out of the 5 weeks. One-way ANOVA test showed that there was a significant difference in total leaf area and above ground biomass for both palmeri amaranth and lamb's quarters between the 3 CO2 treatments every week, except for week 4 in palmeri amaranth. So in all my graphs, they show lines fitted to the raw data set with just the points representing the mean value for each week, just to give you an idea of what each week looks like for the species. This graph shows us that there's clearly a difference in growth pattern over time between the lamb's quarters here on the bottom and the palmeri amaranth here on the top. The rapid growth of palmeri amaranth could indicate a much more rapid rate of maturation than the lamb's quarters, which was supported by the flowering of palmeri amaranth in weeks 4 and 5 under the elevated CO2 conditions. The non-elevated CO2 conditions did not show any flowering by the end of the experiment. So for total leaf area, the two-way ANOVA test showed that there was a significant interaction between the species and CO2 level, indicating that potentially whether the plant is C3 or C4 could affect that how it reacts to elevated CO2 levels. So generally, you can see a difference between the total leaf area over time of the lamb's quarters down here and the palmeri amaranth up here. However, there's an interesting, almost seeming decrease in palmeri amaranth leaf area between weeks 4 and 5. It's possible that lamb's quarter and palmeri amaranth growth was restricted due to competition for light in the closed quarters of the chamber. This might have led to less of an increase in lamb's quarters growth than expected for a C3 plant. Palmeri amaranth was noticeably leggier in the elevated CO2 conditions, indicating that more resource allocation was put towards stem tissue over leaves. This makes the very high leaf area of the control palmeri amaranth in week 5 here really interesting. These plants had a lot more branches with lots of smaller leaves compared to the elevated conditions, which had fewer branches but larger leaves. Also, like I mentioned, by weeks 4 and 5, the palmeri amaranth had begun to flower. This allocation of resource could explain the lack of significant increase in leaf area over the last couple of weeks, while the non-flowering control group was able to continue to allocate to leaves. I think that further study into the effects of CO2 specifically on flowering and reproduction would be interesting for both these species, especially because palmeri amaranth is dioecious. I also decided to look at the leaf area ratio to get a better understanding of how the relationship between leaf area and biomass changed over time. Palmeri amaranth in the control chamber started out with a much higher ratio than the other plants and had a pretty drastic increase by the second week. As you can see here, the lamb's quarters, the pink, yellow, and turquoise lines generally has a less dramatic change in leaf area ratio over time, while the palmeri amaranth has a more continually steep gradient. This aligns with the increased stem tissue observed in elevated CO2 palmeri amaranth. It's possible that with elevated carbon resources, the palmer was able to allocate more growth towards outcompeting the other plants for the limited resources like light, which could explain the increase of stem tissue, which would decrease the leaf area ratio. So in summary, the data suggests that in response to elevated carbon dioxide conditions, palmeri amaranth leaf area will initially increase, but seems to plateau over time, while total biomass increases. While lamb's quarters leaf area and total biomass both increase. In comparison, the C3 weeds total biomass increased less over time, and the C4 weeds total leaf area initially increased more than the C3 before heading for a plateau, while the C3 plants indicate a slower but more steady increase over time. Generally, when we think of C4 plants, we consider them as operating under a more maximized carbon dioxide intake and processing than C3 plants, and therefore don't necessarily expect them to have such a reaction to elevated CO2 compared to C3 plants. But this experiment does show that C4 weed growth is still impacted by elevated CO2, specifically in leaf area, especially in early growth. A broader weed leaf canopy could deprive young crop plants from receiving necessary light required to become economically viable. So it is important to understand these effects. And the study leaves plenty of room for further investigation as listed on the screen. I do plan on looking at nitrogen uptake, having that processed in the lab soon. Also be interesting to look at how other effects of climate change like temperature and precipitation interact with the elevated CO2 levels. And I would like to thank Dr. Dilly and her graduate students for their time in supporting me. And I appreciate the opportunity of the RAIN project and K-State for hosting this. Any questions?
Thank you, Megan. Questions for